immunology is kind of a nuanced subject. Hey, you want to grab one of these? Do me a solid. Thank you. There you go. That's okay. I still get paid. I'm just kidding. Huh? Previous material? I wouldn't worry about it all that much. Honestly, I would pay more attention to what we are doing right now. Unless you're like, I would recommend if you're between 80 to 90% good on what we're doing right now, then feel free to go over the past material. I mean, like, you guys know how to do viable plate count, right? Then those are going to be 2 questions, so, yeah. I don't have the handout with me, so I don't know. But it should be fine. You guys have the whole weekend and then Monday, and then it'll all be over. So do you guys have any questions before I begin? I know we still have a good 30 minutes or so, right? I don't know what time it is. 21. Okay. So what I plan to do today, I'm really just sort of going to go through immunology and intro to bacterial genetics and things like that. And if time permits, I'll try to go over some of the host-microbe interactions. Don't forget about that. That will also be on your final in totality. So those are the 3 big PowerPoints that will be on your final. And then you have the 10 extra questions. My PowerPoint is 144 slides, so I hope you guys are ready to have some fun. What's that? Yeah, I'm gonna try my best. I think the biggest one probably that most people are having trouble with is the immunology one, because I found that material is so difficult to present because it's also interconnected. You don't know what to talk about first. If you talk about something first, then you leave something out, and then you have to bring it up later. So that's probably what we're going to spend the most time on today. I also found that your book was pretty helpful. So if you want to use the book, then I personally felt like using the book. I am going to use PowerPoint first. Yeah, I was going to do it right now. There it is. She said not to use the PowerPoint, but I found that for slides where I would read them and then just be really confused after reading them, going back into the book and finding the information kind of made me feel more satisfied and more comfortable. Of course, it's up to you. But yeah, if those of you who just got in, if you didn't grab an evaluation form up there, you don't have to do it now, but just make sure, pretty please, to fill one out before you peace out. The number is 76. talk to you. Hey, if you guys didn't get an evaluation form on your way in, you don't have to do it right now, but like you just have to get one towards the end and fill it out for me. Do me that solid. That would be great. Thank you. They're up here if you want to get them now and just get it out of the way. I just need them by the end. Or you can just wait. Hi. I'm good. How are you? I'm actually really, really tired. But I'll do my best. Huh? Yeah. Oh, yeah. Thank you. I'm sorry I made you come up here. Ooh, my bad. Sorry. So we have like 7 minutes left before we start. For those of you who just came in, I have it's going to be pretty much like all of my other reviews. I have a PowerPoint prepared. I'm pretty much only going to go over new material. If time permits, I'll try to go over some of the host and microbe interactions. But mainly we'll be talking about immunology and intro to bacterial genetics and molecular biology. Do you guys have any questions for me before we begin? Because I'm just going to go off because I got 144 slides to get through in 2 hours. What's up? Have I seen it? No, but I took this class last semester, so the average for my class's final was like 40, was in the 40s. Yeah, average. Like average raw score before the curve. So, like, you know, it's on the same level of difficulty. You just gotta take it seriously, man. It's nebulous. You can't rely on the curve all the time. But I don't mean to scare you, but I find that if you assume things are going to be more difficult, I'm sorry? Yeah, like 40. It wasn't like 40. It was like 46 or something. Something like that. It wasn't very high, you know. But if you assume something is going to be more difficult than it actually is, then you end up doing better. So maybe that's a good thing. Are there any other questions? Okay. Oh my God, I don't even remember. I don't remember. I don't think so. I don't remember. Maybe. Maybe she'll be benevolent this year. I think you guys actually did better than my class last semester, if I remember correctly, which I probably don't. Hi, guys. Hi. Hi. Good, how are you? Hey. Oh, it is going to be recorded. You'll be able to watch it later online. But you can if you want. If that's your vibe, that's your vibe. You might want to in case there's any kind of error, because it's possible that things might happen, and I can't guarantee that things will go 100% smooth as far as the recording goes, so I would do it just to be safe. Mm-hmm. Hey. Yeah, they're all right here. Here you go. I write a really long review. Oh. Good review. Thank you. Thank you kindly. Again, for those of you who came in, you're going to have to fill out an evaluation form for me. So So they're all up here. By no means do you have to come up and get them now. Just by the end, if you could do this for me, that would be great. The number that we're going to be having is 76. So 76. Again, up to you now or later. I would just be really happy if you did this. Pour moi. Sergio, how's it going? Yeah. I'm sorry? Yeah, actually, what I'm going to try to do is upload it via Dropbox to the SARC website, and I'm not so sure that's going to work. So normally what I would do in a circumstance like that is post it on the Facebook, which is not really fair to people who don't have the Facebook. So if you... in the event that the Dropbox doesn't work and I'm not able to post the PowerPoint, and you don't have a Facebook and you want me to email it to you, at the end of the session you can come up and give me your email, and I'll be happy to email it to you. Again, I'm sorry about that. It's just the website has been kind of wishy-washy lately because we have so much stuff going on it. So yeah. I'm going to try to upload this one and the host microbe interactions one that I did for the last review, in case I don't get to go over it today. So, I'm just going to close. Huh? Oh, no, that's cool. All right, guys, it is now officially noon o'clock, and we're going to get started. So if that's okay with everybody, I'm about to get the ball rolling because, like I said, I have 144 slides to get through in 2 hours. So let's do it. Just a couple things real quick. Again, for those of you who just came in, I'm sorry if I keep on repeating it. For those of you who have been here, if you could pick up a evaluation form, it doesn't have to be now, it can be later, and fill that out for me, that would be great. The number we're going to be using again is 76. So yeah. I had a question earlier, will I be uploading this PowerPoint? I'll try to upload it via Dropbox to the SARC website. In the event that doesn't work, I'm going to post it on the Facebook page. In the event that you don't have a Facebook, please come up after the review and give me your email, and I'll just email it to you. Okay? Cool. Do we have any sort of general questions before we begin? No? Nothing? How do you guys feel about this material? Do you feel like on a scale from 1 to 10, just hold up your fingers. 3, 4, 5, 2, 7. Nice. 5. Okay. 2. That's okay. Don't worry about it. You guys still have 2 days. It's going to be fine. You know, it'll be okay. Just focus and do the work, and you'll be fine. So, all right. I'm going to get started with my business here. Hey, welcome, welcome. All right. So, just a recap for your final exam, it's going to be 100 questions. It's not going to be the standard 50 questions. 90% is going to be from human microbe interactions, which was on your last test. So that's going to be that entire PowerPoint is going to be on the test, not just like little bits of it. So pick out the most important topics and go to it, and study it. If I have time, I'm going to go through some of it today. I doubt I'll have time, though. So it's also going to be overview of immunology and antimicrobials and introduction to molecular biology and microbial genetics, which we're going to talk about today. And it's going to be 10% from previous material. Dr. White gave out a handout in class. I don't know how many of you guys have received that handout. basically outlining the topics that she could ask about with that 10% of the exam. If you're not feeling super good about this part, I would recommend that you don't worry too much about this part. Nah, mean. And again, it's going to be this coming Monday, the 4th at 10 a.m. in the normal classroom. Bring a Scantron, all that good stuff. All right? So just a quick disclaimer, this is not meant to be an all-inclusive study guide. If you wish to truly excel, rely more on class notes and Dr. White's PowerPoints. I am not a PhD in microbiology. This PowerPoint is based on my understanding of the material, and it is meant to assist you, and I just want to help you guys any way that I can. I don't know, I just thought I should throw that out there. Seems like a lot of the SI leaders are doing that. So, the review. Hi guys, welcome, or bye if you're leaving. All right. Is there a question? No? Okay. So what we're going to do, we're going to go over immunology and antimicrobial agents first. Then we're going to go over molecular biology and bacterial genetics. Following, we'll be going over host-microbe interaction. Again, if time permits, I doubt that it will. So feel free to ask questions. Either I or your peers will try to help you. If you feel comfortable with the material covered today during our review, feel free to go over the topics that Dr. White suggested from previous chapters. That's more of a pragmatic tip because, of course, and Bracken said this in class, we want you guys to be like, oh yeah, you have to know everything, but sometimes you have to prioritize, right? And I know that I've been prioritizing a lot lately. All right, so that's it. I'm about to begin. Are there any questions before I get started? Nothing? We're good to go? Ready to learn? Ready to have fun? Again, evaluation forms are up here. You can get it before or after. Doesn't matter to me. I don't care. If you just fill it out, that would be great. All right, so beginning with overview of immunology. And I organized my PowerPoint based on Dr. White's PowerPoints, and I also included some supplementary information from the book. So beginning with virulence versus resistance. This is kind of a bridge from the last chapter, host-microbe interactions, right? So we understand virulence as invasiveness and toxigenicity. And of course, to see more details on that, you have to go over the last PowerPoint. And resistance is defined by acquired and induced immunity as well as natural resistance. So I tried to make this section as clear as possible, but since immunology is such a nebulous part of our course, it's very difficult to organize the information in a way that makes it most clear to you guys. So just bear with me, just stick to the end, and I promise it'll all be clear once we get through all the topics. At least that's how it was for me. So for acquired/induced immunity, we have humoral immunity and cellular immunity. Again, humoral immunity is antibody-mediated, and cellular immunity is mediated by cells like T cells. And these are not a total dichotomy. There can be some bridging. So with that said, natural resistance and innate immunity is the body's built-in ability to recognize and destroy pathogens or their products. And that has to do with phagocytes like macrophages, mechanical barriers, and I put a question here, how can pathogens break through our body's various mechanical barriers? And that requires you to go back to host microbe interactions. So an example of that would be hyaluronidase. Does anyone remember what hyaluronidase does? It breaks down what? Hyaluronic acid, cell cement, something like that. So just a refresher, keep that in the back of your head as we go through this. Another component of natural resistance are chemical factors. So that has to do with interferon, lysozyme, and fatty acids, among others. So going on, and I didn't cover everything in here because I skipped some of the stuff that I feel like you guys could just read and get. So I put some important stuff here. So chemical factors and natural resistance. You have your fatty acids and your bile salts. And I talked about this in SI sessions in the past. Keep in mind that your fatty acids are effective against gram-negative on the skin surface, and bile salts are effective against gram-positive in the gastrointestinal tract. Ooh, there's a big dragonfly in here. Do you see him? There he is. There he goes. All right, sorry. So that's going to be located in the gallbladder, liver, and intestines. Interferon, again, is active against viruses. Lysozyme is effective against gram-positive more so than gram-negative. Why is that? Does anyone know why? LPS layer, maybe? Ring a bell? And what does lysozyme do again? Exactly. What does it do? All right, If you don't remember what it does, I highly recommend that you figure it out. We also have phagocytins. There isn't too much information in the book or online about this. I attached an article that might be of interest to you. So basically all you have to know about this is that it's a bactericidal substance. The pathogen. Complement proteins can also form a pore. So basically they serve two purposes. They can help with phagocytosis. So as the phagocyte is attempting to engulf whatever pathogen, the complement can aid in that, or it can just form a pore. It depends. We didn't go really too much in depth with it, so I don't think that it's ridiculously important. So again, a little more information on complement. Complement proteins are bound by complement receptors called C3 receptors, C3R, which are found on the surface of phagocytes like neutrophils or macrophages, resulting in enhanced opsonization, which is basically like the rendering of the pathogen to phagocytosis, the vulnerability, if you will. So you might recognize this from your slides. I felt like I needed to go a little bit in depth with it just because it was there. So after the antibody binds to the antigen, the complement proteins are attracted to the pathogen. They may lyse the cell immediately or enhance phagocytosis by binding to C3R. FCR is just a receptor that binds to antibodies, right? So here's the C3R receptor, here's the complement, pathogen, pretty basic stuff. Any questions so far? No? All right. So here's some microbial and cellular factors in natural resistance. So of course we went over this. Some microbial factors include normal flora, competition. Recall that if a patient is treated with antibiotics, they're more susceptible to a fungal infection. That's because our antibiotics are effective against all the bacteria in our body, including the normal flora. And the fungus can be an opportunistic pathogen. And we also have cellular factors like phagocytes. So we went over a couple of immune cell types in class, and I kind of broke them up into myeloid precursors and lymphoid precursors because I felt like that was the easiest, kind of outlining that general hierarchy picture that will be on the PowerPoint. We'll look at it later. So myeloid precursors can generate granulocytes, which include neutrophils, mast cells, eosinophils, and phasophils. The neutrophil, which was mentioned a little bit in depth in the PowerPoint, is a phagocyte, an actively motile granulocyte containing large numbers of lysozymes, and it has a segmented nucleus. So just, I recall questions basically asking about the general structure of a neutrophil and that, it's an actively motile granulocyte. Just keep in mind some basic structural properties while you're studying. Okay? We also have monocytes, which are circulating precursors of macrophages, which is another phagocyte. It can ingest and destroy most pathogens and foreign molecules that invade the body. They can also initiate adaptive immunity by presenting antigens to T lymphocytes. And if I'm reading this right now and you have no idea what I'm talking about, hang in there. Once we get through all of the immunology stuff, hopefully it'll all come together. Okay? So they can develop into macrophages or dendritic cells, which are both phagocytes. And then we have our lymphoid precursors. And from those we can generate T cells, which mature in the thymus, or B cells, which mature in the bone marrow or the fetal liver. Fetal liver was added by Dr. White in the PowerPoint, so it's pretty important that you should probably keep that in mind while you're studying. Okay? So here's the hierarchy I'm talking about. So they all come from the bone marrow stem cell. You have your myeloid, you have your lymphoid. Depending on where the lymphoid matures, it can either be a B cell or a T cell. B cells can then differentiate into plasma cells or memory cells, which are not shown here. And then your myeloid precursors, your monocyte, your granulocyte, and basically if you can read this, then you're good to go. This is an important chart to understand. So once the phagocyte has ingested the pathogen, how can it destroy the pathogen, right? So this is a very important slide. I guess every slide is an important slide. So following engulfment, the phagocyte can basically brings the pathogen into what is called a phagolysosome, and these contain toxic oxygen compounds, and we learned about some of these in past PowerPoints. So the ones that you need to keep in mind are hydrogen peroxide, superoxide anions, hydroxyl radicals, singlet oxygen, hypochlorous acid, and nitric oxide, right? And it's not enough for you just to know, oh, hydrogen peroxide, superoxide anion, blah, blah, blah. please know the molecular formula for them as well, because very easily we could ask you a question, or Dr. White could ask you a question that refers to their molecular formula. And why I put, here kind of in, I italicized this, hypochlorous acid. A lot of people mistake it for hypochloric acid or something like that. Know that it's HOCl, and nitric oxide is just no. Okay? How are the pathogens then killed? Via oxidation of their cellular components. Are there any other ways to kill pathogens? Within the phagolysosome, you may also have lysozyme, which is very common. Do we now know what lysozyme does? Did anyone figure it out? What does it do? What's up, Mike? It has something to do with that. It cleaves the beta-1,4 glycosidic bond, right? So keep that in mind. We have our proteases, our phosphatases, our nucleases, and our lipases as well. Okay? These are all enzymes. So, of course, Mother Nature always has her bag of tricks, and pathogens can counter phagocytosis via various means. Dr. White took the liberty of including a couple of different examples that I think that you should know and understand. Like Staphylococcus aureus employs carotenoids and coagulase. So carotenoids basically quench singlet oxygen, and coagulase, which you learned about in human microbe interactions, can create a clot walling a growing population off from phagocytes. So if we create a clot, we can have our population grow, and then we can release something that will dissolve the clot. What dissolves the clot, again? What enzyme is that? Does anyone know? Isn't it like fibrinolysin? Is it fibrinolysin? Am I just messing with you? Who knows? So keep that in mind. Again, this is all information that you have to know. And then another example is Mycobacterium tuberculosis. These guys can actually grow and persist inside macrophages. Although it's not in the PowerPoint, I also included that Mycobacterium tuberculosis can use their cell wall glycolipids to absorb hydroxyl radicals and superoxide anions. Okay, so keep all of this in mind. Pathogens can also employ leukocidins, which kill white blood cells, and they make up, pus is basically, most of it is dead white blood cells, or lysed white blood cells, right? And pathogens that are pus-producing are called pyogenic. So I'm trying to build bridges here between host microbe and immunology. So the two main species that we have to know for that is Staphylococcus aureus and Staphylococcus pyogenes. So pyogenes is pretty easy to remember. Pyogenic, pyogenes. We also have bacterial capsules, which can prevent adherence of phagocyte to bacterial pathogen, and the infamous M protein on Streptococcus pyogenes, which can alter the surface of the bacterial cell and inhibit phagocytosis. The reason why I'm kind of butthurt about this one is because I didn't remember it during an exam, and I got that question wrong. So I don't want you guys to get that question wrong, too. Did she ask about M proteins in the last exam? No? She might do it in this one. I don't know. I was just really upset last semester about it. So continuing on, what are the functions of a phagocyte, right? So obviously, phagocytosis, pathogen destruction, antigen processing via which MHC complex? Class 2. And we're going to go over MHC in a little bit. And like I said, it's really hard to present this all in kind of a 1, 2, 3 kind of way because it's more like a web. So in addition to phagocytosis and antigen destruction, pathogen destruction, they can also be antigen processing. So via MHC class 2 or class 1, they can activate T cells, which T cytotoxic cells can target T cytotoxic cells can cause cell lysis, and T helper cells can cause inflammation, in addition to B cell activation, and we'll talk about that later as well. So again, T cytotoxic cells cause lysis, or rather, a better way to say it would be apoptosis, not lysis, by perforin and granzyme release. Perforin forms the pore through which granzyme can enter and cause apoptosis in the host cell. And TH cells can cause inflammation via release of cytokines and activation of macrophages. So do phagocytes have MHC class 1 or class 2 or both? They have both. Because remember, MHC class 1 is present on all nucleated cells? Is that MHC class 1 or MHC class 2? 1, okay, very good. So keep that in mind as we discuss this more. This is an interesting slide. I'm not really going to go through it, but it may help you to read through it, kind of put everything in context. So I included it in here anyway. I also included this. So, and we're going to go over naturally acquired and passive and active and all that good stuff. But I found this online. So you have your adaptive immunity. It can be artificially acquired or naturally acquired. You have your active and passive for both. So if you read through this, it may help put everything in context for you. So I'm not going to go through it now because we'll go through it in a little bit. And again, here's another slide. Immunity, innate immunity, adaptive, natural, artificial, passive, active, passive, active. So if you present it like this, it may help things be a little more digestible for you. Okay? So some details on induced immunity. It basically requires prior contact or exposure to the antigen. Humoral immunity, of course, as we mentioned before, is mediated by antibodies, which are produced by B lymphocytes, which can then differentiate into plasma cells, which can produce antibodies en masse or memory cells. Cellular immunity is mediated by cells, like T cells and natural killer cells, which we'll talk about in a little bit. Okay? So here's a nice slide on T cells. And again, if you haven't really studied yet and a lot of the stuff is kind of confusing you, again, it's all going to come together in the end. Just be patient with it and don't give up hope. So again, we know our T cells come from the lymphoid precursor. We know they mature in the thymus, hence the name, T cells. And we'll go into more details following a discussion on matrix compatibility complex. So our T helper cells contain the CD4 coreceptor, right? And the CD4 coreceptor is often, a lot of people think it's on the cell presenting the antigen, but it's actually on the T cell, the CD receptor, CD4, CD8. T helper cells are also associated with MHC class 2, which is associated with B lymphocytes, macrophages, and dendritic cells, which are basically all dedicated antigen-presenting cells. So we have our two types of T helper cells. We have Th1 and Th2. Th1 can produce cytokines that stimulate macrophages, that increase phagocyte activity, and promote inflammation. Th2 can activate B cells and thus antibody production. Okay? And so that's one way that cells can be involved in humoral immunity. On the flip side, we have our T cytotoxic cells, which contain the CD8 coreceptors, which are associated with MHC class 1, which is also associated with all nucleated cells. Cells displaying a foreign antigen via MHC class 1 are killed by T cytotoxic cells. So basically what this is saying is that T cytotoxic cells associated with MHC class 1, and they have the CD8 coreceptor, are basically associated with cells that have an intrinsic infection. So an example of that would be like a viral infection, right? So if your cell is infected by some kind of virus, then you can assume that, MHC class 1 will come into effect, T cytotoxic cells will come in and kill the cell presenting the antigen. Because it's a lot easier to kill a cell with an intrinsic infection versus kill the infection within the cell. So might as well just kill the whole thing. Does that make sense? Yeah, I'm not as eloquent as I should be today, and I apologize for that. We know that T cytotoxic cells will release perforins and granzymes, and as I mentioned before, perforins create the pore that granzymes can go through, and granzymes will cause apoptosis, which is programmed cell death, cell suicide, essentially. Very morbid. I included a slide on natural killer cells. If you noticed in your PowerPoint, Dr. White doesn't mention this until the very end. I found it a little bit helpful to mention it now, but it's up to you. Honestly, it really depends on your taste, on how you want to organize this material. So natural killer cells are a class of lymphoid cells. They kill tumor cells, virally infected cells, bacteria, fungi, and various parasites. They're nonspecific, and they are involved in the innate immune response. Okay? They're involved in target cell killing. They initiate apoptosis in the target cell by the release of perforin and granzymes, so that they're related to T cytotoxic cells in that way. They also produce cytokines, TNF and the granulocyte macrophage stimulating factor. It might be important for you to know these two words, or class of words rather. They don't use toll-like receptors. I know we didn't mention it yet, but bear with me. They function as a protective mechanism, and their action takes place in the interval before the more powerful adaptive immune response comes in. So you have your natural killer cells coming in doing the work, and then the adaptive immunity will kick in.